The first speaker is Daniela Rhodes. Daniela joined the lab as a technician in 1969. Later on, she registered as a PhD student, and I had the pleasure of being her internal PhD examiner. Daniela just reminded me, the other examiner was Beverly Griffin, so it was an all-women team, which is quite unusual, certainly was in those days. Daniela went on to become a group leader at LMB, and she clocked up a total of 41 years at LMB before the retirement age made her leave and go on to other things. Daniela is a fellow of the Royal Society and a fellow of EMBO. Daniela.
I'd like to also personally thank Kathy for producing such interesting, wonderful book. And also, it's also wonderful to be in this lecture theater and see so many women. It's not usual. So I thought for today, rather than concentrating on the science I've been done, I'll tell you my story, because I think I didn't have a usual career in science, and it might help some women to realize that you can have a career in science not following the normal straight path. So my scientific journey. So I was actually born in Italy and left Italy when I was at infant school. My parents emigrated to Sweden, and I have listed my personal versus my professional career. So I was born in Italy and moved to Sweden at the age of 10 and a half. And in 1965, I actually had a gap year in Cambridge because I didn't know what I wanted to study. I was ready to apply to art school because that was my passion, but happened to have won the mathematics prize every year at the school I was at. So whilst I was in Cambridge, I actually met my husband-to-be, and I decided to get my act together, went back to Sweden, and I did chemical engineering and graduated in 1969. I graduated in chemical engineering, married 2 years later, and took the boat to England. And my husband lived in Cambridge. And I knew I had to work because he was still studying. And we actually lived with my parents-in-law, who lived down the road from the old LMB on Hills Road. And I saw an advertisement for a technician job at the MRC Laboratory of Molecular Biology. I actually didn't know what molecular biology was because I'd done chemical engineering, and in the 60s, it was still pretty unknown. But I did borrow a book from the library, and I got an interview with this man called A. Klug, and I had no idea who he was. Anyway, I turned up at the LMB, he interviewed me, and it was a very strange interview, possibly due to the fact that I was wearing a miniskirt. It didn't occur to me that it was not appropriate to go to an interview in a miniskirt. But he asked me, what did you enjoy most when you're doing chemical engineering? I said, physical chemistry. And then he said, okay, you start Monday, basically. So within 4 weeks of arriving in Cambridge, I had to work at the LMB as a junior research assistant. Aaron hired me to work on they were working on the crystal structure of t-RNA, which was the first crystal structure actually of nucleic acid. I remind you that the DNA structure was derived from fibers, DNA fibers. It wasn't crystallography. And so Aaron hired me, but immediately put me to work with Brian Clark, who was in cell biology, and who was, I think, he was a new group leader. And I must say, I'm grateful to Brian because he was actually never in the lab and seemed to travel a lot. And he had this habit. He wasn't nurturing you. He would come back to the lab after his travel and say, have you screwed it up again, guys? And he wasn't discriminating between men and women. He would talk to us in the same way, whether we were male and female. So basically, from the very beginning, I had to try and figure out by myself how anything worked, including, how do you run the centrifuge? But I was surrounded, at the LMB with amazing people. I was in 305. Linda Chapman was Simpson. I was grinding up tobacco plants to make tobacco mosaic virus. Joe Butler and Tony Durham were trying to figure out assembly of the tobacco mosaic virus, Uli Lemley and Jake Messel invented Lemley gels in 305 in Cambridge, which is, Lemley is the highest cited biochemist in the world due to the Lemley gels. And, I had Fred Sanger at the end of the corridor, Cesar Milstein, and there was this open door policy. So if you didn't know something, I remember talking to Fred, was Nick translating DNA and it wasn't working. At 10 o'clock at night, I'd see Fred at the end of the corridor. I said, oh, Fred, I have this problem. He said, Klenov is somewhat sequence specific. Tickle your DNA with DNS1 and it will work. And it did. So it made me take responsibility for my work. And although I was a technician, I would say I could probably not have had a career anywhere else except at the LMB, because first of all, science at LMB was almost like an art form. It was very exciting to be here. Once I started understanding a bit about science, I did realize that it was a fantastic place. And at LMB, I discovered science and great workplace and great mentor. And how did I do it? I already said I moved to Cambridge because I'm married, but accidentally found this work sometimes. Okay, great mentor and great workplace. My mentor was Aaron Klug. And I was also fortunate that once I found my feet, Brian Clark left LMB and then Aaron started talking to me, which then changed things. When I fell in love with science, I think when I saw my first electron density map of t-RNA, electron density map might look a mess, but the ones for nucleic acid, anybody can immediately recognize the structure because you see the planar bases, you see the zigzag ribose phosphate backbone. And actually, what made me want to have a career in science? I was going along very well as a research assistant. They sent me off to international meeting. I had a son, and I decided now it's time to take responsibility for my career. And so I started my PhD after my son was born. And it's a picture of the old LMB. And actually, I think we were working at this end, but I think this is a picture before the end extension was built. I was working in 305, which was at the top underneath the canteen. And this, I don't know when that extension was built, but sometime between 62 and 69. And then, of course, the new LMB. And at the time, I don't know if it still says so on the website of the LMB, but the aim of the LMB was of the laboratories to study the structure and function and synthesis of large molecules of biological importance. And I think we would all agree that LMB shaped the life of young researchers, whether they were male or females, and including mine. And Max Perutz was the founder of the LMB. And together, of course, he had this amazing talent of recruiting the best people, and he recruited Sidney Brenner, Aaron Klug, Francis Crick was his graduate student, and Fred, who already had won Nobel Prize and then went on to get the second. So together they created this, I would say it's a philosophy about how to do science. And Max's philosophy LMB was spot talented people and support them to grow their own research. Rank does not count, only ideas. Open culture, always ask questions. Make decisions based on science. And this philosophy and lack of hierarchy is empowering, and it's empowering for both men and women. So I personally never felt I was treated differently because I was a woman, but there were a few surprises, and I forgot to say earlier, when Aaron offered me the position, he said, and at the MRC, we pay men and women the same. And I thought, what is he talking about? I was coming from Sweden, and I thought it's obvious that you pay men and women the same, okay? And also, having gone to a high technical school to do engineering, I was actually used to being surrounded by men, because I think in an engineering school, only like, even in Sweden, only like 5% of the students were female. So it wasn't so odd to me to enter into this man's world. I knew how to deal with men. So as a technician, I published 13 papers. And actually, I highlighted, I have 3 single author papers from my time as a technician. And this shows the LMB. The idea then, at least, was if it was your idea, it was your paper. And Aaron, of course, would read the paper because, you know, they didn't want to ruin the reputation of the LMB. But making the point that, you know, I was made to write earlier my own paper and recognized, I mean, they were not big ideas, but they were my papers. And so my papers, my name was on the major paper, including the first paper actually was a Nature paper. And after I've only been here for 2 years, I've been at the LMB, you know, the structure of t-RNA, the structure of the nucleosome, and so on. And I reconstituted actually the first nucleosomes with homogeneous DNA and crystallized them already in 79. And then, so these 2 projects really shaped my career, but I want to point out something. So in 1974, this is the structure of Finland, 3-angstrom resolution. I want you to see the authors. The females were marked out by having the name written out, you know, Jane Ladner, Crawford, I know, she was Crawford, Ladner, she had married by then, and Daniela Rhodes. And I said to Aaron, well, if you are a Klug, I want to be D. Rhodes. So in 77, I became, in the next stage, I was D. Rhodes. And actually, later on, I met someone, was introduced to someone, and he said, oh, I always thought you were a man. And I don't know if I should be flattered by this or insulted. So crystal structure, I go through this quickly, so I think I will run out of time. You know, what's really important, the famous adapter molecule, it was incredibly difficult project. It certainly shaped you in terms of, I think I purified 8 different t-RNAs and the crystals didn't diffract. And it was also politics and competition. We were competing with Alex Rich at MIT. But through that project, I discovered the beauty of structural biology. And that's t-RNA. And here is the picture of the people working t-RNA, John Finch, Aaron Klug, you know, the metal models, Richard Box and Brian. Notice I'm not there. And then because I had grown all the crystals for t-RNA and found some derivative, after Roger Kronberg had defined the nucleosome core, and then later had figured out how to produce fairly homogeneous preps of nucleosome core from natural sources, Aaron said, would you like to have a go at crystallizing the nucleosome core? And actually, it was incredibly easy. I got crystals the second time I tried. And here is actually EM picture of first the nucleosomes lining up. After a couple of hours, you set up the crystallization, and here is the hexagonal packing on nucleosome in the crystal. And so it was after this project that I said, you know, oh yeah, and I want to show this because this is one of my favorite pictures of Aaron. You know, he has people who know him that look, he always smiled when he talked about something he enjoyed, and he's holding the nucleosome and talking about the nucleosome. So this is, and also I like to show this to young people. You know, this is pre-computer graphics. We had to build a model, which is bolts of wood, which we painted, you know, where the histones were actually based on Jim Thomas's cross-linking work. We knew the relationship within the histone, right? And then the plastic tubing is DNA. And actually my thesis was born out of the problem that we knew before we solved the structure, there had to be 2 negative supercoil around the nucleosome, but experimentally we could only find 1, and it became known as the linker number paradox. So my thesis was on figuring out what's the helical periodicity of DNA solution. Turns out, because the linking number is the sum between the twist and the rise. And in fact, it's different. The DNA twists up to bind to the histone octamer. And then, so now we go to, I finished my PhD, and at that point, Sidney Brenner was the director of the lab, and he said, go away, you never get a job here. And Aaron said, no, stay. And then I got tenure. I went straight from technician to a tenured position at the LMB. And getting tenure was quite memorable because actually I went down to London with Kyoshi Nagai, who sadly passed away. And the two of us had the same committee. Both of us got tenure, a tenure position that day. And then I stayed at the LMB until I retired in 2011. And I had an offer from Singapore. And I went to Singapore, first as a professor just to work on my science. And then they asked me to set up an Institute of Structural Biology. So I'm the founding director. We set up an institute. Well, it contained 85 people. And within that, we had all the structural methods, NMR, crystal. I even bought an X-ray generator because there wasn't one working in Singapore, and all the synchrotron stations were really far away. And we set up cryo-EM. And the person who actually set up cryo-EM is Sarah, who was here at the LMB with me for many years. And this is where cryo-EM, well, this is, we set it up within material science, the EM unit that built new facilities. And then, yeah, and I retired from Singapore about 2 years ago. And really these two projects, t-RNA and the nucleosome, shaped the rest of my career. I went on to work on how protein recognize DNA, how high order chromatin structure, and how histone mark modify chromatin structure. We actually were some of the first people with Tom Check to discover proteins that bind to G quadruplexes, and then went on to find the first in vivo evidence for G quadruplexes. And that field has become enormous now. I just come back from a meeting where there were 250 people. And then my last 20 years, I worked on telomeres. And I just finished with one picture of our last paper, which came out in Nature just a month ago. And you see, a good project lasts a lifetime. I mean, I started with the nucleosome. Also quite nice. My first paper was in Nature, and my last one will be in Nature. So thank you very much.
I was wondering if you had any advice for people at the beginning of their career in science.
I think the only advice, if you want to be in science, you really have to be passionate about it and be convinced that if you do your best possible work, it's going to work out.
I mean, there'd be a lot of time for discussion of that sort of issue at the panel, I think.
Yeah.
Yes, one more question.
Thank you for a beautiful description of your career. I have a quick question about how was your transition from technician to PhD? Did you see any change of behavior? Or?
None, because I was already working independently as a technician. I was allowed to publish papers, a single author. And actually, someone at MRC head office said to me, when they came to LMB, they said, I always wondered how you did your PhD at the same time as you were a technician. And I never let her in. She was an administrator. I never let her into the fact that the two were exactly the same as far as I was concerned.